All right. So week one, we are going to go through 2 PowerPoints this week. So I will share my screen here and then we can get going on that. Just share my entire screen. There we go. Okay. So you should be able to see the first PowerPoint is going to be the biggest part of the info for the week. It's the bacterial genetics and metabolism. So a lot of this is probably going to be carryover if you've ever taken a biology course, a general micro, or a anatomy and physiology type course. And of course, we don't always remember everything from those. But let me know if you have any issues seeing what I'm showing. Nope, I don't want to do that. Are you seeing the instructor side or the big display side? I think you're just seeing the display side.
I see it says bacterial genome and objectives.
Yep. So, okay. 1 second. Let me redo this here. Hold on. I know what I have to do. Sorry. First weeks. There. Why are you doing that? Oh, it doesn't. I want it to. Oh. Okay. I'm just going to leave it alone. Apparently, me and this are not going to cooperate today. So hopefully you can see this. I'm just going to keep it in this view because when I go into the presenter view, it doesn't want to work for me. So bacterial genetics and metabolism, there is objectives for the PowerPoint and for the week, as always. I'm not going to read those to you. You guys know how to read. So we're just going to first start out with prokaryote versus eukaryote. And this is just a reminder coming back to A&P. Like I said, we don't always retain everything. So you might hear these words once in a while in the beginning here as we're describing our microorganisms. And so the difference between the two really comes down to what they have. So prokaryotes are very small. So that's going to be a lot like our bacteria. They don't have any organelles. So think like no, mitochondria, that kind of thing. They do not have a true nucleus. They're unicellular. They do have a cell wall. And then they reproduce asexually, whereas eukaryotes are going to be your complex things like animals, plants, fungus are all considered eukaryotes. So they will have organelles, they will have a nucleus, they're uni or multicellular, and they're much larger than the prokaryotes, and they can reproduce asexually or sexually or both. But so that's kind of the main difference between the two. So bacteria are going to be in the prokaryote world for microbiology, and then fungi are eukaryotes, just more complex. But both will have ribosomes because they have to make proteins, and ribosomes help us make proteins. They have cell membranes a lot of times, DNA, of course, and cytoplasm. So they do have some similarities there. And then I did have a nice little image representation because, it's a lot easier if you can see something with your own eyes. So again, the prokaryotes are more bacteria. Sometimes our bacteria have flagella to help them swim. Some do not. Not all bacteria do, but they can have that flagella as well there. But you can just see like eukaryotes are much more complex. They have a lot more of those organelles and things inside there. All right. So when we talk about microbiology with microorganisms, the word itself just means like you can't see it with your human eye. Fungus, you can, like molds, you can, but overall, we need like a microscope to really see and identify these items. So that's why they're called microorganisms. So there are 4 main areas: bacteria, parasites, fungi, and viruses. And when we say fungi, that means both molds and yeasts. So in a micro 2, at the very beginning of microbiology 2, we talk about parasites and fungi more there. So we'll reserve that toward micro 2. We talk, of course, about bacteria throughout both micro 1 and 2. Viruses, we don't do a lot with in micro. This is kind of one of those areas that's a little bit immunology and then a little bit microbiology. So you might hear some brought up toward the end of micro, and then you might hear about viruses more in immunology as well. With you think of hepatitis, HIV, those are viruses, things like that. So it's kind of one of those topics that gets spread out for our curriculum a little bit. All right, so again, bacteria are prokaryotes, they're unicellular, and again, they lack that nucleus, mitochondria, endoplasmic reticulum, and other organelles. Parasites are going to be considered eukaryotes along with the fungi. And so there's a reminder of fungi or eukaryotes. Again, I don't test you on what's a prokaryote, what's a eukaryote, by the way, on tests. This is information. This is just setting us up to learn about these things going forward. And then viruses are actually the weird guyotes. Again, they are the smallest. Can't see them with a normal microscope, a typical microscope in the lab. They're neither prokaryote, eukaryote. They do have DNA or RNA, not both. They're based either on DNA or RNA, one or the other. They're technically not even a living thing because they need us to live, a cell to live, something else to live, and to replicate. So they're just kind of its own little genre out there, if you will. All right, so when we think about naming and classifying these organisms, especially bacteria, we have a whole microbial taxonomy system. So that means the classification, nomenclature, which is how do we decide the name, and then that allows us help with identification as well. Now, the beautiful thing is as science advances and we learn more, we rename things because we've learned more about those bugs. So you will see where bacteria changes names. And there'll be ones that like we always have called this name and all of a sudden they learned more about it and then reclassified it. So then they renamed it and they realized, oh, it doesn't quite belong over here. Now it belongs over here. So we're going to rename it to show that. And this happens quite a bit. So it's kind of one of those things we have to always keep up on the names, and it can be hard. So there might be things that I'm so used to calling a said name and now it's changed. So it's one of those things you almost get used to hearing it both ways. You know, it's never simple. We never like to have one name for anything. But again, by having this whole classification and name system, that way worldwide, wherever you go for a lab, for clinical micro, you know what you're talking about. You're all talking about the same bacteria. It's not called something different in Europe than it is over here. So that really helps. All right, so you probably learned back in high school biology, the whole classification or taxonomy system going from kingdom through species. So kingdom is the largest group and then division, class, and it kind of gets narrower and narrower as it goes down. In the world of bacteria and fungus and parasites, we're dealing with genus and species levels mainly. So we're getting really honed into the system or the classification that they exist in. So again, this is all just organizing them because they share certain features. They are genetically alike. They look alike. Morphology kind of is the way they appear. The different tests that they react to is similar, that kind of thing. They grouped them together for that reason. Okay, so again, when we deal with genus and then species, we're talking again about very similar physiologic genetic traits. You can even go further down and get even more specific into subspecies. So that's even a subgroup of that species. And then you get even more specific in what we call biotypes or serotypes. And if you ever work in a larger, like reference microbiology lab or like the State Department micro lab, you will deal with serotypes for sure. They are not dealt with in every hospital micro lab, depending on the size of them. But some organisms definitely have to deal with serotypes. So it's just even more specific characteristics. I think about Shigella is one of those, and that's a popular food poisoning organism. That deals a lot with serotypes. That gets sent off to the State Department lab or specific reference labs to serotype which Shigella serotype is causing this issue. So they take that into account. All right, so again, genus and then species. So all of our bacterial species will belong to a genus, and then the species of that genus gives it further genotype and phenotype characteristics. So we don't really talk anything higher than a genus in microbiology. So we're always talking about genus and species. So that is how we name them, is a two-name system. So when you see them named, it's the first part of that name is their genus. The second word of their name is species. So if I were to say Staphylococcus aureus, that's probably a bacteria you're familiar with or you've heard of at least. Staphylococcus is the genus, and then aureus is the species. And the way it's written is anytime you write out organism names, they have to be italicized. The genus is capitalized. The species is lowercase. So the way that this example here that I've kind of highlighted is written, that is exactly how a bacterial name should be written. That is something I want you to remember going forward. You'll have some case study projects to do this quarter, so you'll want to put that in those case study projects. Plus, I think I have a question on this on a test. So if I ask you how to properly write the name of an organism, it's both words are italicized, the genus is capital, the species is all lowercase. So that is the proper way to write it. All right, so a lot of this kind of seems repetitive with what I've already said. Identification, how to identify bacteria, especially our microorganisms, is just by their features. So we have different tests and stains and things like that that we use. And then we know like different genus and species will react certain ways, and that's how we look for what it is. So we can use genotypic characteristics and phenotypic characteristics. So you hear that a lot. Genotypic just sounds like what it is, genes. When you think genes, you think DNA. So it's all based on their genetic DNA makeup. And especially as science advances, we get more and more ability to do this. And so that's the world of what we call molecular. That's molecular biology. Molecular testing is always dealing with DNA stuff, genetic stuff. So micro is getting to be crazily advanced compared to where it used to be. Of course, in our education realm, we are still sticking with the basic tests because you need to know those tests for your boards. Once you get working in a real micro lab, you are going to be amazed at the technology they have out there, but we still have to learn the basics. So the fun stuff will come later in like clinicals or, you know, working wise, if you work in a micro department, it gets to be really fun what they can do. But genotypic is all about DNA. Phenotypic is what is the morphology, like how does it look on an agar plate? How does it stain? What is the stain reaction? Different tests that we are going to run in your micro lab. You get to play with these tests. They're quite fun. Is it positive? Is it negative for that test? And you put all of that together and then you can figure out what you have. And so that's what we'll be playing heavily with in our labs is the phenotype stuff, because again, you have to know it for boards. They ask that. They will give you a question with all these test results and you'll be like, oh, it's this organism because I know those results, that kind of thing. So that's the difference between genotypic and phenotypic, but both are used to help identify the organism. All right, so a little bit into the world of bacterial genetics. This is not very exciting stuff. A lot of this is kind of, again, background information. I'll make sure to tell you if you need to really know something, but overall, the genetics of bacteria, so it's ability to be viable, adapt, multiply, cause disease. Bacteria are like people. They want to survive. They want to do their thing just like we want to do our thing. So they are going to do everything they can to keep surviving and thriving. So the way that three major aspects of their genetics, they have structure and organization. So their material is organized. They can replicate and express that genes just like us. And then they have mechanisms by which they can exchange that genetic information with other bacteria. And that kind of helps play into the world of mutation and adapting as they need to. So just like you probably have learned at some point in biology A&P, nucleic acid structure, we have DNA and RNA. DNA is made up of a sugar, phosphate, and a base, so they're nucleotides. And then these nucleotide chains are put together in a specific sequence, and that is our genetic code, if you will. So this is all just information background again. And you can read, as you know, as you can probably tell by now, I don't read slides. I discuss them. So pause this at any time you're listening and read through it yourself if that helps. But again, this is not, I'm not going to test you on what are the DNA nucleotide bases. We're past that. But if you did want to know, they're here. They're again for DNA, it's two strands together. They're complementary to each other. So they have the adenine and guanine bases that pair and the cytosine and thymine bases that pair. For a second there, I had a blank. See, I blanked it out myself. So they pair together and they make that double helix for DNA. All right, some terms that hopefully you kind of are familiar with, but again, a reminder, a gene is that DNA sequence and it codes for a product. Like we have different proteins that we have to have in our body. So we have genes that code to make each of those proteins for us. Same with bacteria. A genome is all the genes together in an organism. And then again, that genome is put into what we call chromosomes. All right, so there are also other little things that are called plasmid. They're kind of like if you think mini chromosome. They're not really a chromosome, but they still belong inside a bacteria and they still have some genetic stuff. They have DNA. So we call them mini chromosomes, but are they truly a chromosome? Probably not. They're non-chromosomal element, as you see. So they're actually small circular DNA that some bacteria can contain up to like 500 of these in their cell, in their little bacteria unicell. So each bacteria varies on how many plasmids they have, but it's another way that they have DNA. And there goes my dogs. Okay, hopefully they don't bark for too on here. But anyway, so again, they can carry DNA in their normal chromosomal DNA makeup, but also in plasmids. So they're just not as stable as true chromosomes. So there's a little picture down here to kind of give you an idea. But so plasmids are kind of unique here for DNA, carrying DNA in bacteria. I would know that term a little bit, just know the basics of that term. Okay. All right. So again, replication, all bacteria want to multiply by cell division. So they're going to make 2 cells from their one parent cell. And as they do that, they replicate their entire genome to do that. And they give that genome to those daughter cells. So there's different stages of being able to replicate listed there. Again, I am not testing on this. This is just background information. It's similar probably to what you've learned at some point. They then have to express that genetic information. So they have to use it. So they have transcription and translation. Transcription is going to convert their DNA into RNA. And then translation is all about protein synthesis. So that is amino acid sequences. You have to put amino acids together to make a protein. You probably learned that in chemistry as well. And so that is what they're doing with translation, is making those proteins. So in order, again, for a bacteria to survive, they have to have all that gene expression and protein synthesis. And so they will regulate this whole process as well through those 3 levels, again, information. All right, as I said earlier, though, bacteria are a lot like us. They want to survive in any way they can. And sometimes that means that they have to achieve genetic diversity. They have to change their genes at some point. So the way that they're built right now, they can't have any real genetic diversity if they didn't have the ability to exchange with other bacteria or mutate somehow. We wouldn't even have evolution. We wouldn't have the ability to grow and evolve without genetic diversity. So they do this through mutation, genetic recombination, and gene exchange. One of those 3 ways will help them, give them new genetic diversity to allow them to adapt to a new environment and keep thriving. Because otherwise, if you think about it, like, we could have wiped out bacteria if they could never change and never mutate. Our medicine would have wiped them out at this point by now. But because they can adapt and change and mutate, they keep us on our toes. And they're like, oh, well, now they're resistant to this drug, and now we have to find new drugs to treat it. And it's problematic, but that's what they're doing, is trying to survive. So mutation is just, they alter their genes. They can alter their genes somehow. So they just change the sequence of that gene and it becomes a slightly different gene. And now it's not the same. So that's mutation, is it's just an alteration of their sequence in their gene. Genetic recombination is where they take a segment of their DNA and they exchange it with a different segment of DNA from another cell. So they swap it. They swap DNA segments. And they kind of recombine that way. So the new DNA segment comes in and puts into where the other one was. And that will change up their genes. And then gene exchange is they can actually physically exchange DNA. That it could be that there's DNA floating out there that they take in, that they're like, oh, we're going to grab that free DNA that's just in the environment and have it. And so they have taken up that free DNA that's in the environment. They can take DNA from 2 bacteria that come together for recombination. So that will kind of exchange genes there. And then they have conjugation, where they can kind of like 2 living cells, they require contact. And then again, they're having movement. So it's pretty similar, each one of these, but basically you get the idea. They're putting new DNA in place of the other stuff and swapping out. So those are the 3 main ways that they get genetic diversity and allow them to keep surviving. So, and then of course, for bacteria to keep thriving in general, they have to have their ability to function. So you have to have energy for the bacteria to thrive. Just like people's bodies need energy, the bacteria need energy. So fueling, they have to create stuff to help them survive. They got to be able to have biosynthesis and then assemble what they create. So I kind of put in a picture example. of this just because I think it helps. Again, this is not something I test you on. It's just, if you ever go deeper into like a master's degree level, PhD level, you really get deep, deep, deep into this. We're keeping this big picture because we have already enough stuff to get through. But again, fueling, basically this is what is saying we need energy. So they take energy from sugars, amino acids, fatty acids. That's just like people. Humans do the same thing. We use those items as well to give our cells fuel. So they're going to take those items to get fuel. And these pathways here are like this pentose phosphate pathway, glycolysis, gluconeogenesis, all that. You probably learned those words in clinical chemistry. These are all pathways that we use. Okay, how can we take these sources and convert them into energy? So these are the pathways that will convert them into energy. So once they get this energy, now we are going to start creating stuff. We have ribosomes, cell wall creation, synthesis. We got to upkeep that cell wall. We have virulence factors. Sometimes bacteria have capsules, so you got to keep that going. You have toxins that are being produced. These are all like defense systems of a bacteria. So they need the ability to do all that. So that's what they're using the energy and pathways to create and uphold. So that's just kind of a strong overview. These get into it more. Again, these are all background information. So I'm just going to kind of skip through with, it's basically what I discussed. If you hear the word anabolic processes, that means they're assembling, they're making things. Catabolism is breaking down something. So like they might take in light and chemicals and then break down that chemical into substances they can use. And then anabolism is the opposite. They're going to then use those building blocks to create something. So it's just, they need both sides. They need to break down certain things for energy, and then they are going to build up and create something with that energy. So that's that whole process for metabolism of a bacteria. All right, so now getting into some more of the goodies that we need to know for what we're going to move forward with. Structure and function of bacterial cells. So again, I said earlier, prokaryotes, eukaryotes. So bacteria are the prokaryotes. We already discussed that. Parasites, fungi, and plants, animals are all eukaryotes. So with the prokaryotes, again, it does have a cell wall, and the cell wall is made of a substance called peptidoglycan. And you are going to hear that word because the stains that you're going to use, the main stain in Microlab is called the Gram stain, and it makes a difference how much peptidoglycan a bacteria has. So we are going to learn a little bit more on that. So I have that word bolded. Whoops, my goodness. I have that word bolded here for a reason. So we are going to hit that here on the next slide. So that cell wall peptidoglycan is going to become important of what makes up bacteria. All right, so peptidoglycan is basically short peptides. So it's right in the word peptido. So it's made of these glycan strands that are cross-linked by short peptides. The word itself tells you what it is, essentially. So peptidoglycan just adds strength to the cell wall. It protects it, the whole cell, the whole bacteria itself, and then it helps with cell division. So it has an important role there. So when you look at, like, say this is a bacteria down here, the red inside is the cell membrane, and then the outside is the actual cell wall. So that cell wall is where the peptidoglycan is based. So the red here on the bottom is the cell membrane. Not all bacteria have cell membranes. Those that do would look like this. And then you have peptidoglycan on top of it to give it that extra cell protection and strength. So when we look at Gram staining, which is what you'll start with right away in Microlab, is all about Gram staining, because that is the first thing we do when we think about identifying organisms, is we stain them. What Gram stain reaction? Because we have what we call Gram positives and Gram negatives. So based on the way that they take up stain, they'll either be a Gram positive organism or they'll be a Gram negative. So Gram positives will be purple. Sometimes you might call it a really dark blue. I just say purple. It's a very dark purple. So that is a Gram positive. So if you look under the microscope and you see it's a dark purple color, you call it a Gram positive. If you look under that microscope and it is a pink to red, that is Gram negative. So that is the first step, is just determining is it positive or negative. So, and that difference in what stain reaction or the color it is because of how much peptidoglycan was in that cell wall. Once you determine the color reaction, then you look at the shape, because bacteria can be different shape. They can be cocci, which are like round balls. They can be coccobacilli, which are more ovoid. So think oval, you know, it's kind of a combination between the round and then the rod, which is bacilli. So they can also be bacilli, which is rod. There are spiral shaped bacteria, spirochetes. We will talk about those in week 10 of this quarter. But you don't really see them a lot, and you don't really test them the same way that you test normal bacteria. So they kind of get a separate category, but there is technically a spirochete shape as well as spiral. So when you report out a gram stain reaction, you say gram positive cocci, gram positive bacilli, or you can say gram negative cocci, gram negative rods, gram negative bacilli. Rods and bacilli are kind of used interchangeably. You can call them either way. It's the same thing, bacilli or rod. So that is our gram stain reaction, as we're reporting out the positive and negative and then the shape with it. Because when we start learning our bacteria, we have bacteria that are always gram positive cocci, we have bacteria that are always gram negative rods, and so on. So we have them all categorized that way. And then if you go further into it, you might start to talk about, oh, you have certain bacteria that love to occur in pairs, and they're always in pairs. When I think about it, when we learn Neisseria gonorrhea, you guys have heard of gonorrhea, that is caused by the bacteria Neisseria gonorrhea. They always occur in pairs. They're what we call a diplococci, diplo for two. That is a very big characteristic of them. So when we see that, when we were like, oh, it's a gram negative diplococci, that's one thing it might be. So there's certain features like that that they always have. So we also take that into account. And as we learn the different bacteria, we'll discuss those characteristics at that time. All right, cell envelope is the outermost structure of a bacteria. There also might be that outer cell membrane with it. That's only in gram-negative bacteria. So that is important to remember. Cell outer membranes, only gram-negative bacteria. Both gram-negative and gram-positive have cell walls, but gram-negative only have that extra membrane. So just keep that in mind. It has that extra barrier there to it. Again, for the cell walls, the peptidoglycan thickness makes a difference. Gram-positives have a much thicker layer of peptidoglycan, and they also have something we call teichoic acid crosslinks in it. It really it's a thick layer, and it's really strengthened by those teichoic acids. So gram-positive, thicker layer of peptidoglycan, important to remember. And then there's also, of course, cytoplasm, and a lipid bilayer and all that kind of stuff there for structure. Let's see here. I have an example of what that difference between the peptidoglycan looks like with the membrane. So again, gram-negative bacteria will have this additional outer membrane on top, and then it will have the peptidoglycan layer, and then both have a cytoplasm membrane. So you'll see here, the gram-positives have a much thicker peptidoglycan layer, but no membrane on top of that. So that's kind of the primary difference between the two. And that's why they take up stain differently. So I think next week we get into the actual stain process. So we'll cover this again next week, but just kind of FYI. All right, more components of a bacteria. Cytosol is just the interior world of the cell. Of course, they have DNA, might be existing and also in a plasmid DNA. And then some bacteria can produce spores. Not all bacteria, just some. So when we start learning different bacteria, we'll talk about if they are able to produce spores or not. If you think of one that's fun to talk about, not when I say fun, it's because fun, nerdy stuff, not because real world I don't want it to happen, but anthrax is a spore forming bacteria. That's why the whole thing with like breathing in through envelopes, bioterrorism, that whole thing. So that can occur because it has the ability to make spores. So we'll talk about different ones when we get into it, if they can make those spores. All right, and then nutritional factors. So just like people, bacteria need nutrition to get energy to do their functions. So they get this through different like environmental things. So when we grow bacteria in the lab, we put them on agar, what we call media, to give them that nutrition so that they grow. So in that agar could be either liquid or like solid, like in a plate, and you'll see this in lab. That has different nutrition in it so that they like will thrive. We'll put them in the incubator where it's nice and toasty warm, and then they use the nutrition in the agar to help them grow so that we can look at them and play with them. Fastidious bacteria will be a term that you'll start to hear this week through next week and the next week and on. In a couple of weeks, we'll really have to know that term well. And fastidious just means these are the extra picky bacteria. They don't want just the regular agar. They're not happy with that. It's like kind of thinking about really picky children. Actually, I'm probably picky like this. I want the extra stuff. I didn't want the regular. I'm picky on what I want for nutrition. So we have that agar that's really like going to give them that extra nutrients to be like, here, now take this agar and grow. So there is some bacteria that are called fastidious. So this is, again, a term we'll hear in the next few weeks, especially. They're just a little bit more complex. They want extra nutrients than the normal. All right, and as I said, media can be in broth form, which is a liquid, or in a solid form. So in the broth, the nutrients are dissolved in the water. We can inoculate the bacteria. So you can dip the swab or whatever, your loop, into the broth, swirl it around, put it in the incubator. And then if the next day or the 2 days, however long it takes, if it becomes cloudy or turbid, that tells you there's growth. There is bacterial growth inside that broth. And I have a picture of this on the next slide here. And then agar is in a plate form. It's kind of like jello looking and feeling when you touch it. You'll learn how in lab to streak out your sample onto the agar plate, and then the next day you'll see colonies forming. So this is kind of what it will look like. So in a broth, this very left tube here is not inoculated. There's nothing in it. This is just how the tube looks. So it's kind of clear. Ignore the weird color at the top there. It's just kind of clear, but you can see, look at the difference, what it can look like when it does grow bacteria. This cloudiness or turbidity, that tells us, oh, there is bacteria growing there. So we'll learn more about that. I think in week 3, we'll talk about agars and different types of broths and stuff like that. And then again, when it grows on an agar plate, it will form what we call colonies. So you can, and this is, you'll see this all quarter long, you'll get to play with this. And then environmental factors. So as I said, we put them into an incubator. The incubator is set at body temperature, which is 35 to 37 degrees Celsius. That is standard incubator temperature. That is what most bacteria want to grow at, is your body temp. There is a form, a certain bacteria called Campylobacter. Maybe you've heard of that if you've ever gotten food poisoning. There's Campylobacter jejuni, common cause of food poisoning, especially from chicken, raw chicken. That bugger likes to grow even warmer. It wants to be in a 42-degree environment. It likes it even hotter. So I would remember that, by the way. Campylobacter loves 42-degree temp. Most bacteria love a 35 to 37. And then there's those bacteria that depends on what kind of levels of oxygen and CO2 they have. Strict aerobic bacteria need a lot of oxygen. Anaerobes are the opposite. They want no oxygen. They want to be put into an anaerobic environment inside that incubator. So we have the ability to do that. We can make an anaerobic where it removes the oxygen out of there so they can grow. Then there's a term that's called facultative anaerobes. That means they can grow with or without oxygen. Doesn't matter. They'll grow either way. Capnophiles want increased amounts of CO2. So the term capno, the med term there, means carbon dioxide. Phile means loving. If you ever know that from medical terminology, phile always means it loves. Capno is the term for carbon dioxide. So the word itself, capnophile, means loves carbon dioxide. So in this case, it wants around a 5 to 10% CO2 level and 15% oxygen level to grow. So there are certain bacteria, you'll do this in lab, where we'll put in what we call candle jars to help increase the amount of carbon dioxide, things like that for those. And then you have the term microaerophiles. So micro for small, aero for oxygen. So it just means it wants less oxygen. It wants oxygen to be down around 5 to 10%, and then it will bump up the CO2 to 8 to 10%. So the CO2 with capnophiles and microaerophiles is around the same level. It's going to be the oxygen that kind of varies. Capnophiles can leave the oxygen up around 15%, but microaerophiles really want less oxygen. I would know the difference between these terms. You should know an aerobe versus anaerobe versus capnophile versus microaerophile. They're bolded there for a reason. So. All right, so that is a ton of information thrown at you. Again, the good news is a lot of this first PowerPoint was just some background information. I tried to make sure to point out what was necessary there. So let's go over to our other PowerPoint here, infection control in microbiology. Again, you have some objectives there you can look through. But you have just some stages of microbe to human interaction. You have the physical encounter when you first encounter the microbe and the host, your human. You have colonization on the human surface, their entry, their invasion, they want to spread, and then what's the outcome. So those are kind of the overall stages of what happens. So some terms that you're going to have to know, again, they're bolded. Reservoir. So when we begin learning bacteria, we begin learning where do they like to hang out? So what's their reservoir? What's their place of origin? Some bacteria are always found inside humans. That's where the reservoir is. Some bacteria are found in certain animals, water, certain foods, air, soil. Those are all types of reservoirs. Where are they hanging out? And that's where they are coming from. Mode of transmission, how did you get it? How did you pick up that dang bacteria? That can be through a vector, like an animal, so a living thing, a mosquito, a fly, a gnat, any lice, you know, ticks. Those are all common vectors that will pass you those goodies. Or it could be through what we call a fomite, or which is a non-living thing. So think of like doorknobs, you know, that's a common one, right? Cell phones, like countertops, things like that, that somebody has sneezed on or passed it on and then it didn't get clean and you come along and touch it and you don't realize it and that's how you got it. So that is a fomite, is non-living. So those are definitely terms to know as well, vector and fomite. So that is the mode of transmission. So you can, as a human, get it, of course, from another human. If that human was the reservoir, and then you could pass it to the other human. Examples are a newborn passing through the birth canal of the mom. The birth canal has a lot of bacteria hanging out in it. It's a natural part of it. So maybe they pick up something through while they're being born. Could also get it through different blood transfusions. We've done a pretty good job on the screening all that, but you never know. Sexual contact, of course, and then indirect as well. So like I said, if that person sneezed on a countertop and then you touch it, that's a way to get it as well from a different human. All right, you can also get it from animals through animal bites, scratches, or through a vector that has fed on that animal and then come and fed on you. So these are what we call zoonotic infections. So they are infections that are happening in the human because it's from an animal microbe. So Lyme disease is common on this. Lyme disease is passed by a tick. So that tick can come off like, say, a deer or something else, and then it comes onto you and it passes, you know, that bacteria into you that causes Lyme disease. All right, survival again, as I said before, is the key to a microbe. It wants an environment that's going to fulfill its needs to give it energy to thrive. So it's always searching that environment out, and unfortunately, it loves us. So another term that's for this, colonization, you do need to know that. That is the persistent survival of that microbe on the surface of your body. So that is the very first step it has in like wanting to further cause disease or infection. So it's surviving, it's hanging out. So that's colonization. So of course, us from a human role is our immune system should hopefully play a, you know, hopefully we have a really robust immune system that will take care of it and protect us. You know, the very first line of defense. So if you're in immunology, which most of you probably take this quarter at the same time, the first line of defense is your skin and mucous membrane. So if it's just hanging out on your skin, but you have no wounds, no scratches, no openings, you should probably be okay. You're washing your body, you're washing your hands. So it's hopefully getting rid of it and removing it anyway. And it's not having an opening for it to get into. So your skin and mucous membranes are there. That is your first line of defense, and that should hopefully stop any further infection. But of course, if it does have an opening or possibility, it's going to dig into it. So to establish an infection, the microbe then will first overcome that physical barrier, overcome that skin and mucous membrane. They will do this through altering the environment conditions somehow. They could change the way that your cells are behaving. They could change your normal flora population. So normal flora is the bacteria that you just have as a natural part of you. And it does actually some good work. It's not a bad thing. The normal flora is good. You never want to, you know, whenever I hear people like messing with like their GI system and doing a cleanse, I'm like, you want that bacteria there for a reason. It's there for a reason. So never clean out everything purposefully. So that's there for a reason, because otherwise, once you wipe that out, then it allows something else to come and thrive and be like, oh, there's nothing here to stop me. I can really overcome this and take it over. So that's kind of our normal flora, is our bacteria that lives in us and helps us for a good thing. So we have other non-specific responses. So once a bacteria can overcome your skin and mucous membranes, then your body goes into, like, we have different chemicals we can release. We have different cells that get involved, like phagocytic cells that try to eat the bacteria. We have inflammation that happens. So these are all very immune system stuff. I am not going to discuss this here because you are going to learn this heavily in immunology. So immunology will teach you all about these processes here. So we'll just keep going. Once they get past the non-specific, then they go into the specific response, which involves antibodies. This is where we make antibodies to really defend that off. Again, you are going to learn more about this in immunology, so we'll leave it alone for that. All right, difference between infection and disease. You probably hear these terms used interchangeably. They're actually a little different. Infection is the growth and multiplication of microbes that can cause damage to you. Disease happens when that damage becomes noticeable. It is affecting your organs. It is affecting something, and it becomes very noticeable. So like an infection could be strep throat. You know, that is an infection of your throat area by a strep bacteria. That's just an infection. But there is ways that that bacteria can then go, and it's been known to affect the heart and cause rheumatic fever afterwards. That's now becoming a disease because now it's affected one of your organs. So that's showing how it can go from infection to disease when it causes noticeable changes in different organs of you. So that's kind of the difference there. Virulence factor, again, as we learn about different bacteria, we learn about their virulence factors. This is how much can they cause disease? What can they do to really do a damage? So certain bacteria can produce really bad toxins. Certain bacteria have the ability to have great motility. They have capsules that make it really hard for us to penetrate and break them down and get rid of it. They have ability to have stickiness and form like a grouping of them and like a biofilm layer over them so that you can't penetrate and get them gone. So virulence factors are whatever they can do to live and survive and keep causing infection. So the more virulent they are, the more disease causing they are. All right, so as they invade, they will attach, invade, survive against your immune system, essentially. So we've already discussed all that. That's kind of general info. Again, as I said, they have virulence factors. Some of that might be toxins they can produce. So those are different substances that they release and that they definitely will have an effect on us host cells. A lot of food poisonings act that way. It's actually the toxin from the bacteria that is causing you the food poisoning, where it's basically killing your like GI cells and causing you all that diarrhea or vomiting or both with that infection. And the crazy part is you don't even need the bacteria around anymore. Once it's released all the toxins, if you just ingest the toxins only and the bacteria is not there, it's just the toxin, it will still cause all that damage and cause you that infection with the toxin alone. So there is two types of toxins. Endotoxin is released more by gram-negative bacteria. Exotoxin is released more by gram-positive bacteria. The way that I remember that is I always think of that N in endo for negative. That's kind of how I keep it straight in my brain. All right, and so the outcome of how do you fare once you get this infection and or disease, it depends on your immune system. Were you a pretty healthy person? Did you have a good immune system? If you're somebody that has had leukemia or some cancer or some other illnesses that really make it tough on your immune system, like your immune system's kind of shot because you're already sick with these, then you're going to really be prone to being very ill then with anything else. So it depends on your health. It depends on the virulence of that pathogen, how virulent it truly was. Like I said, there are some organisms that are really virulent, and then there's some that rarely cause any disease at all. And then how fast can you clear it out? And then, of course, prevention is vaccines, immunizations. You'll learn more about that in immunology, but that is one of the best ways to prevent anything is if there is a vaccine for it. Okay, so discussing a little bit about sterilization, disinfection processes, that kind of thing. So sterilization means all forms of that microbe is killed or destroyed. So we have physical sterilization where you have burning through an incinerator. You have autoclaves. Autoclaves are commonly used, especially in a dental office. A lot of times you see them sitting like little mini ones sitting right on the countertops that they use to clean their dental tools. But we also have autoclaves, of course, hospital wise, if you think surgical instruments, radiation using gamma rays. So those are all forms of physical sterilization to kill microbes. There's also chemical sterilization. So there's a bunch of different chemicals. Formaldehyde is probably a popular one you've heard of. And then disinfection is where you're basically killing a lot of the organisms, but not all. It's not as thorough as sterilization. Sterilization was aiming to kill everything. Disinfection is just mainly getting rid of a lot of it. Some things are going to still survive. Like if there's spores, they probably will still survive. So you have physical disinfection, like boiling water. When my daughter was really little, I used to boil water and then let it get back to room temperature, of course, to make her formula, because that way it just kind of help keep that water pretty clean. Pasteurization, UV lights. I know you can buy like UV lights for your phone to clean your phone off, because they say your phones are like one of your most dirty things ever. So people have bought like UV lights for their phones at home to kind of help disinfect them. And of course, chemical disinfectants as well there. So we make different bleach solution in the lab to kind of disinfect and clean off our countertops, things like that. And then, as always, safety, safety, safety. You've heard of this in different classes. So you have your chemical safety, which includes your standard, your safety data sheets. I couldn't think of the first S there for a second. Sorry. Safety data sheets. It used to be called MSDS. Now it's just called SDS, safety data sheets. You also have to have a chemical hygiene plan in every lab. Basically, what do you do if you get exposed to that chemical? Fire safety, which involves, of course, fire extinguishers, electrical safety, if there's compressed gas, that kind of thing. Biosafety is all about like how do we protect ourselves against, infection from the different bacteria, viruses, whatever might be in that person's blood or body fluids. So we always have standard precautions, which means we're treating everything and everyone as if they could have something. So we're always wearing our PPE, our gloves, lab coats. That's the minimum. You might even wear goggles or a face shield, depending on what you're doing. Masks. In the lab world, in the microbiology lab area, we have biosafety cabinets. Every micro lab has a biosafety cabinets. So you'll do an assignment where there's 4 levels of biosafety cabinet, and you have to kind of identify what each level is used for. And then engineering controls are actual like negative air pressure in the lab environment. So it's removing the air out, not blowing air in with a bunch of stuff in it. Then the BSC stands for biosafety cabinet. Again, using the biosafety cabinet. And again, there's 4 classes. Most hospitals are class 2. So most regular micro labs in a hospital are level 2. And then the bigger the micro lab gets, the more it's going to be used for level 3 to level 4 kind of thing. Level 4 is insane. Very interesting, but that's a high level. You're really working with some crazy stuff. And then shipping, mailing, there's a bunch of rules for like if you have to ship and mail out like different samples, organisms to like the state lab or a reference lab for further identification and tracking. There's all sorts of ways that you have to ship and mail it. We are not worrying about that here. That is not something we're going to get into. All right, so that is module 1 PowerPoints. In your course, let me just pull this over for you. You'll also see that there's this little thing in there called Lab Safety Microscopy and Aseptic Techniques. You can go through this and play it if you want. It's just good information. Like if you click number 2 here, you can use this up here. You can read through it at your will. There's little like activities in here to do. Again, here's more parts of the microscope. If you don't know a lot from hematology yet or anything, heme should have really got you familiar with your microscope. It's going to be the same for micro. Again, lab instructor, every week you're going to be playing with your microscope, looking at gram stains. So you work with your lab instructor on that as well, and they will help you. But yeah, there's a microscope game. So safety data sheet. So if you click on this, this will kind of flip through. So again, it used to be called MSDS. Now it's just called SDS, just so you are aware. but click through that. It's good information. I just wanted to highlight that that is there as well. So, all right. Otherwise than that, that is all I have for this week. I'm going to stop the recording.